Cell, the unit of life. Hi, dear students. I am Dr. Vetri. I am basically an ENT surgeon from Tamil Nadu, India. I have many national and international teaching experiences. But for the first time, I am just preparing this small study material in biology. The first chapter which I am going to talk is Cell, the unit of life. It's a very interesting chapter. This is basically made for the plus 1 and plus 2 students who are appearing for competitive exams. Go through this video study material and enjoy. Mitochondria. The number of mitochondria depends upon the physiological activity of the cell. Mitochondria differ in size and shape and can take a shape of sausage or cylindrical. The diameter is approximately 1 micrometer and the length is approximately 4 micrometer. Mitochondria is also named as powerhouse of the cell or ATP mill in cell, cell within cell. Most busy and active organelle in the cell is the mitochondria. Semi-autonomous cell organelle is the mitochondria. Endosymbionts of the cell is mitochondria. So coming to the structure, it is stained by Janus Greenbee. Mitochondria is double unit membrane, referred with the vacuole which is single. The outer membrane is smooth and inner one has plenty of folds which is called as crista. The metabolically active mitochondria, the number of crista is higher. So the inner compartment is filled with dense homogeneous substance which is called as the matrix. Both membrane are separated by a space called perimitochondrial or intermembrane space. Many electron carrier cytochromes are arranged in a definite sequence in the inner membrane of the mitochondria which form the electron transport system ETS. Inner membrane is studded with pinhead particles called oxysomes or elementary particles referred to elementary bodies of mycoplasma, or F0 to F1 particles or ATP synthetase. These particles first described by Fernandez Moran. The head of oxysomes of F1 is concerned with oxidative phosphorylation, formation of ATP by energy of oxidation. Mitochondrial matrix have enzyme for Krebs cycle which is aerobic respiration. Besides this enzyme, matrix have a complete protein synthesis apparatus. So it synthesizes ribosome which is 70S, DNA, few RNAs and enzymes. Because of this, mitochondria is called as semi-autonomous cell organelle. So single, double or circular naked DNA are present in the mitochondrial matrix. Understand where you have the circular DNA which is guanine cytosine rich. Biogenesis of mitochondria is how the mitochondria is formed. New mitochondria arise from division of pre-existing mitochondria by process of binary fission. So it is also of endosymbiotic origin from prokaryotic cells. Coming on to the next topic is the plastids. Plastids are found in all plant cells and in euglenoids. They are observed with the microscope. They are large. They have pigments classified into chloroplast, chromoplast and leucoplast. The chromoplast is a fat-soluble carotenoid pigment like keratin and xanthophyllus and others are present. And this color gives the color of yellow, orange, red color to the part of the plant. Chlorophylls are either absent or occur in very less amount. Chromoplast occurs mainly in pericarp and petals. Red color of tomatoes is due to the red pigment lycopene of chromoplasts. Second thing is the chloroplast. The chloroplast contain chlorophyll and carotenoid pigments which respond for trapping light energy essential for photosynthesis. Leucoplasts are colorless. You have the amyloplast which store protein is present in potato. Eleoplasts store oil and fats. Aleuroplasts store protein. Pigment and lamellar structures are absent in leucoplast. Generally occurs in non-green and underground plant cells. Coming on to the numbers, shape and size of chloroplast.
The majority of chloroplast of green plants are found in the mesophyll cells of the leaves. The number varies from, it can be just one chloroplast per cell in case of chlamydomonas of green algae, or it can be per cell you have 20 to 40 chloroplast in the mesophyll cells. These are lens-shaped oval spherical disc or even ribbon-shaped. The length is approximately 5 to 10 micrometer and the width is of 2 to 4 micrometer. The structure of the chloroplast, it has got a membrane like mitochondria. It is also double membrane. Outer membrane opening called porins. So coming on to the structure of chloroplast, you have to understand the outer membrane, inner membrane, the stroma lamella, granum, stroma, ribosome, starch granules. And you have to see exactly where the location of thylakoid is there located. Go through the diagram. Compounds of the stroma include thylakoids. So in stroma has thylakoids, enzymes, it has got nuclear material DNA and ribosome. So A is thylakoids. In the stroma, number of organized flattened membranous sacs. These are called thylakoids in the stroma. They are arranged in the position of piles called grana, right? Or the grana which are called the thylakoids. Each chloroplast contain about 40 to 60 granum. Stroma lamellae or Friet channel or stroma thylakoids are flat membranous tubules connecting the thylakoids of a different granum. So it is long. Okay, see, go through the diagram. The membrane of the thylakoid encloses a space called lumen. Chlorophyll are present in thylakoid membrane. A photosynthesis functional unit located in the thylakoid membrane contains of about 250 to 400 molecules of various pigments of chlorophyll A, chlorophyll B, keratin, xanthophyll, etc. is called as quantosome. The enzymes, it has got various enzymes of carbohydrate, that is enzymes of Calvin cycle or Dock reaction and enzymes for protein synthesis. The DNA, the stroma contains small double-stranded circular DNA molecule. Ribosome of the chloroplast are smaller 70s than the cytoplasmic ribosome 80s. It is also considered as a semi-autonomous organelle. The main function is photosynthesis. It helps in the biogenesis from proplastid or from binary fission of pre-existing plastids. Origin is endosymbiotic origin by the cyanobacterium. The next topic will be the ribosomes. The ribosomes are the engine of the cell. They are the engine of the cell. Ribosomes are the granular structures. So invented by George Pradade. They are composed of ribonucleic acid or RNA and protein. So ribosome is a combination of ribonucleic acid along with the protein. It is not surrounded by any membrane. Understand? It is membraneless. Except mammalian RBC, all living cells have ribosomes, both prokaryotes and eukaryotes. Ribosome are the smallest cell organelle. Ribosome also called as organelle within organelle and protein factory cell. So, type of ribosome, you have got the eukaryotic ribosome and the prokaryotic ribosome. Eukaryotic ribosome of the type 80S occur in the cytoplasm of eukaryotic cell. Prokaryotic ribosome is 70S type, occurs in the cytoplasm and associated with the plasma membrane of prokaryotic cell. Their size is 15 to 20 nanometer. 70S ribosome is also present in mitochondria and chloroplast of eukaryotes. S is the Swedberg unit or the sedimentation rate. It indirectly is a measure of density and size. Each ribosome is composed of two subunits, that is larger and smaller. Here the 80S has 60S plus 40S and the 70S is equal to 50S plus 30S. Magnesium ion is essential for binding the ribosome subunits. Magnesium ions form ionic bond with phosphate groups of rRNA of two subunits. Several ribosomes attach to a single messenger RNA and form a chain called polyribosome or polysome. So you have the ribosome, you have got the messenger RNA, you have got the transfer RNA and you have got the ribosomal RNA. So the three types of RNA are there. So already I told you the rRNA is considered the connection of two magnesium ions, whereas the continuous chain of messenger RNA forms a chain called polyribosome or polysome. So both the ribosomes are different. Coming on to the cytoskeleton. The elaborate network of filamentous proteinaceous structure present in the cytoplasm is collectively referred to as chromosome. They maintain the shape of the cell. Then microtubules are composed of protein called tubulin, size of 25 nanometer approximately. In plants, microtubules are often associated with the cell wall. Probably these transport cell wall material to the Golgi body outside the cell. In animal cell, during cell division, these microtubules form spindle fibers. Next is the microfilaments. So these are contractile protein. Microtubules is different from microfilaments. It is the microfilaments are composed of contractile protein. You got the actin, which is concerned with the muscle contraction, and the microtubules and microfilament are part of cytoskeleton base of skull. The next topic would be the cilia and the flagella. So you have cilia, small structure which work like oars, like a boat. The movement of either the cell or of the surrounding fluid. The flagella are longer, respond for cell movement. It is an electron microscopic study of the cilia flagellum show they are covered with the plasma membrane. Their core is the axoneme. It possesses a number of microtubules running parallel to its long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of an axonemal microtubule is referred to as 9+2 array or 9 doublet plus 2 singlet. Please go through the diagram. You understand the outer inner dynein arms which has ATPase activity. Understand the peripheral microtubule, the doublets. Understand the interdoublet bridge or the AB linker which is formed by a nexin protein. Understand the central microtubule, the plasma membrane, the radial spoke, the central sheath, and the bridge which connects the central microtubule. So these are basic things which you understand with the diagram. The central tubules are connected by bridges and are enclosed by central sheath which is connected to one of the tubules. There are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilia and flagellum emerge from the centriole-like structure called the basal bodies or blepharoplast. So where does it emerge? The cilium and flagella? The answer is the basal bodies or blepharoplast. The basal bodies are different from the elementary bodies. Coming on to the cilia and flagella. Now first understanding the cilia. The cilia are small in size, 5 to 10 micrometer. Flagella are long, up to 150 micrometer. The number of cilia per cell is very large, whereas flagella is very few in number. Cilia beat in a coordinated manner, which is called as a pendular movement. The flagella beats independently, which is called as an undulating movement. The cilia take part in the locomotion, attachment, feeding and sensation, whereas the flagella is involved only in the locomotion. The next topic would be the centrosome and centrioles. While going through this chapter, kindly understand that centromere is different from centrosome and centrioles. Okay, centromere is seen in the chromosome. Okay, so they will talk about that later. So centrosome is absent in higher plants. Centrosome containing 2 centrioles is called as the diplosome. So the centriosome containing 2 centrioles is called diplosome. It is located just outside the nucleus and lie at right angle 90 degree to each other. Each centriole is surrounded by amorphous pericentriolar materials. Centrioles are membraneless. So once I say membraneless, you should understand that ribosomes are also membraneless, right? So ribosome is membraneless and the centrosome and centrioles are membraneless. The cylindrical structure, it's a cartwheel structure in transverse section. Centriole contains 9 evenly placed peripheral triplet fibrils of tubulin. These triplets are linked with the help of AC linker. AC linker is adenine and cytosine. So it is amino acid. So AC linker. It has nothing to do with air conditioning. The central part of the centriole is proteinaceous and called a hub, which is connected with peripheral triplets by radial spokes made of protein. It is 9+0 arrangement. Centrioles are self-duplicating units. It can divide by itself. Function in animal cells, centrioles play an important role in cell division. That you'll understand when you go through the mitosis chapter. By arranging spindle fibers between two poles of the cell, the location of centrioles during cell division decides the plane of division. The plane of division is always at right angle to the spindle. Centrioles form the basal body of cilia or flagella. Understand? Centrioles form the basal body. You got the diagram of the flagella, isn't it? So the basal body is formed by the centrioles. Coming to the microbodies, there are many membrane-bound minute vesicles which contain various enzymes that represent both plant and animal cell. These are the microbodies. It is the peroxisomes or uricosomes. These are found both in plant and animal cells. Peroxisome contain catalase enzyme. It's concerned with the peroxidase metabolism. Catalase degrade the hydrogen peroxide into water and oxygen. In plant, peroxisome occurs in cells, green tissues, and concerned with photorespiration. That is the glycolate pathway. Peroxisomes are also involved in beta oxidation of fatty acids. Then you have the microbe, the glycosomes, glyoxysomes. Glyoxysomes occurs only in plants, especially in fatty seeds, castor seed, groundnuts, etc. Glyoxysomes are considered as highly specialized peroxisomes. It is a variation of peroxisomes. Glycolate acid cycle takes place in glycosomes. The cycle converts fats to carbohydrates. You will understand that later. So, the important points include several ribosomes are attached to a single messenger RNA form polysome. In germinating seed, fatty acids are degraded exclusively in the glyoxysome. Peroxisomes with chloroplast and mitochondria perform photorespiration or glycolate cycle. So, the most important topic now is the nucleus. Nucleus is also a cell organelle. It is described by Robert Brown. The nucleus is stained by the basic tie called as acetocaramine and the name given by Fleming as chromatin. Nucleus is a double membrane bound dense protoplasmic body which controls all cellular metabolism and encloses genetic information of the cell. Generally, eukaryotic cells contain at least one nucleus, but nucleus is absent in mature phloem sieve tubes. You should know where it is absent. Nucleus is absent in mature phloem sieve tubes and elements and in mature RBCs of mammals it is absent. It is dikaryotic in paramecium, that is there are two nucleus in paramecium, and multikaryotic cells are also known. So when there is multikaryotic we will see that. The structure of interphase nucleus. Interphase the nucleus is a stage where the nucleus is not dividing. So that is the best stage to study the nucleus. The nuclear membrane it's got a nuclear membrane called karyotheca. It has got a nuclear matrix which is also called as a nucleoplasm or karyolymph or karyoplasm. It has got a chromatin net and it also has a nucleolus. The other names of nucleolus are the little nucleus or the ribosome factory. So ribosome factory is the nucleolus. So understand the diagram of the nucleus, the outer nuclear membrane, the inner nuclear membrane, both of them how they form the nuclear envelope, the perinuclear space, how the ribosomes are attached, the nuclear lamina, the nucleolus and the chromatin, the nuclear pores, the arranged position of the cytosol on the rough endoplasmic reticulum attachment to the nucleus. The nuclear membrane has got a nuclear envelope, which has got two parallel membranes. It has got a perinuclear space. The outer membrane usually remains continuous with the endoplasmic reticulum. Understand? The outer membrane usually remains continuous with the endoplasmic reticulum and also bears some ribosomes on it. And then it has got an opening called the nuclear pore, which is guarded by octagonal discoid structure called nucleoplasmin protein. So the nuclear pore is guarded by a octagonal discoid structure of nucleoplasmin protein. The inner side of the inner nuclear membrane is called nuclear lamina. The structure is formed by filaments of lamin protein. So nuclear lamina is made of lamin protein. Second thing is the nucleoplasm of karyolymph. It's colloidal. It contains nucleotides, nucleosides, ATPs, proteins, and enzymes. The chromatin net and nucleus are components of nucleoplasm. So, understanding the chromatin net, the term given by Fleming, interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, which is embedded in the nucleoplasm. Chromatin net is mainly formed of DNA and histone protein complexes. So, chromatin net is made up of DNA and histone protein complexes. Chromatic fibers contain genetic information and they are condensed to form chromosome during cell division. So, when do they condense? They condense only in the stage of cell division. Other times they will be like a net in the form of chromatin net. So, condensation occurs during cell division. During different stages of cell division, cells store structured chromosomes in place of nucleus. Okay, so when the cell division takes place, the nucleus is replaced by structured chromosomes. Chemically, chromatin consists of DNA, RNA, and histone proteins. Okay, initially what we saw was the contents of the nucleoplasm. So, we saw that the nucleoplasm contains nucleotides, nucleosides, ATPs, proteins, and enzymes. So, now chromatin contains DNA, RNA, histone protein. See, it has a basic protein rich in arginine and lysine and non-histones. So, the histone proteins are basic proteins which are rich in arginine and lysine, and it also has non-histone proteins. Chromatin net has two types of chromatins. It is called the euchromatin and heterochromatin. So, the euchromatin is genetically more active. It is lightly stained and diffused part of chromatin. Heterochromatin is darkly stained and thick and condensed part of chromatin. It is genetically less active than inactive protein or inactive chromatin. Coming on to nucleolus, the nucleoli are spherical and membraneless structure. It is contains the rest of the nucleoplasm. So, understand, it is a site for active ribosomal RNA or rRNA synthesis. So, nucleus is that's why they say it is a ribosomal factory. Nucleus usually attached to the chromatin or chromosome at specific site called nuclear organizer region. NOR, nucleus attached to the chromatin at specific site called nuclear organizer region. Nucleus is called ribosome factory of the cell, which is seen already. Large amount and more numerous nucleoli are present in cells actively carrying out protein synthesis. So, coming on to chromosomes, the next chapter. At the time of cell division, chromatin metal gets condensed to form chromosomes. Thus, chromosome is highly condensed form of chromatin. Chromosomes are not visible during interphase. Chromosome can be best studied at metaphase because the size of a chromosome is the shortest in the metaphase. The shape of the chromosome is best studied at anaphase stage. The number of chromosomes in a gamete is called genome. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 or 23 pair of chromosomes. Based on type of chromosome, the base of position of the centromere, refer to the diagram. Telocentric when the centromere is terminal or located in the tip of the chromosome. Acrocentric when the centromere is subterminal located near the tip. Metacentric when the centromere is attached at mid of the chromosome. Submetacentric when the chromosome located near center or midpoint of the chromosome. The ratio of the long arm to the short arm chromosome is called arm ratio. Arm ratio is maximum for acrocentric chromosome. Structure of the chromosome. So, you have got the chromatid, you have got the centromere, you have got the satellite, you have got the secondary constriction, you have got the telomere. So, the 5 important points concerning the chromosome are these 5. So, and go along with the diagram, understand the diagram, see the kinetochore, see the primary constriction of centromere, see the secondary constriction or NOR, see the position of the satellite, see the position of the telomere. So, the diagram represents double chromatin chromosome. So, chromatid is best seen at the metaphase stage of chromosome. So, it is in the region of the cell division of 2 cylindrical structures called chromatids. Both sister chromatids are joined together by a common centromere. A chromosome may have single chromatid or two chromatid. So single chromatid in anaphase and telophase, two chromatid in the early cell division of prophase and metaphase. So you know the first stage is prophase, second stage is metaphase, third phase is anaphase, and the last phase is telophase. So in the first two phase, you got two chromatid, and the second two phase you got single chromatid. Okay, when you go to the mitosis, you'll understand better. Single chromatid consists of single long threaded DNA associated with histone. Non-histone proteins and RNA are also present. Coming to the centromere, each chromosome at prophase or metaphase, it consists of two chromatids. Both chromatids of the chromosome are joined or connected by a structure called centromere. At centromere, two protein discs are present, which are called kinetochore. Kinetochore constitute the actual site of attachment of spindle to chromosome during cell division. So directly the spindle does not get attached to the centromere. It gets attached to the point or disc called as kinetochores. At the region of centromere, the chromosome comparatively narrower than the remaining part of the chromosome. Thus, it is termed as primary constriction. Please refer to the diagram. And second thing is you got what is the second. Third point is the secondary constriction. Besides, other constriction also occur. These are non-staining and found at constant location. Okay, so secondary is also known as NOR or nuclear organizer region. Represents the 13, 14, 15, 21, 22 chromosomes in human. For a satellite, part of the chromosome remains after the NOR is known as chromosome satellite. Telomere, chromosome have polarity and polar ends of chromosomes are known as telomeres. So you have got 4 in if there is 2 chromosomes, right? Telomere prevents fusion of one chromosome to other chromosome. Telomere is rich in guanine base. Enzyme telomerase synthesizes telomere part of chromosome, which is ribonucleoprotein. Telomeres of chromosome becomes shorter during aging process.